the same lab, one floor up right there. It used to be the fluid mechanics lab, and now it's the Hydsopolis microfluids lab. Actually, Jeff and I had the same advisor, me for a master's degree and Jeff for his PhD, and that was Forbes Dewey. Back at that time, Jeff was looking at turbulence generated in a stenotic artery, and he's gone a long ways from that to becoming probably the leading expert, I think, in cell rheology, and having developed a lot of the concepts that have now become sort of dogma in terms of, what is the mechanical, the structural behavior of a cell, and then how does that change over time, and what factors account for it. And then more recently, Jeff has moved into done some really interesting work looking at cell population collective cell migration. That I think you're going to talk about a little bit today.
Yeah.
And that's why I wanted to point out that movie that Ray had. I hadn't planned this, but it actually works out very well, I think, that Jeff is speaking now. So it's a real pleasure to have Jeff here, and thanks for coming across the river. Yeah. We'll pay all your expenses.
Yeah. I walked. Yeah, it's a pleasure to be. It's funny, as Roger said, it was in the late 60s and early 70s that we were together upstairs, and you wind up changing fields, but you keep bumping into the same people in these different fields. So it's funny how that works. And it's also, it was kind of interesting to hear Roger summarize my work, calling it dogma, because I've spent the last 20 years trying to overturn dogma, and now I get a big fat bullseye. So you guys can show everything I've done is wrong. But that's what science is, right? The truth is an approximation, and it's always changing. So that's a good thing. So I am going to talk about collective cell migration, but I'm not going to start there. I'm going to start with something else, and I'm going to get into it. And it's going to actually dovetail, I think, very nice with the previous lecture, which I actually love, because I don't know anything about embryology, but the things we're doing on collective cell migration kind of butt up to that, and a lot of the ideas will resonate, I hope. So I want to start this way. So I'd like everyone to just take a big, deep breath when I count to 3. Ready? 1, 2, 3. Now, didn't that feel great? Think about it. So your lung is probably the most extensible of all organs, and when you did that, you stretched every adherent cell in your lung, the endothelium, epithelium, smooth muscle cells. You stretched them all, but if you went all the way up, you stretched them by about 20% of their length. And just when you're doing just quiet tidal breathing, like you're all doing now, with every breath, you're stretching them by about 4% of their length. And it turns out that those stretches are big enough that they have a dramatic effect on the cytoskeleton. And I'm going to mention this, and I'm not going to go into it much. The cytoskeleton mechanically is actually much closer to being an elastic solid than a viscous fluid. We're going to come back to that. Most biologists think about the inside of the cell as being a chemical liquid factory, but it's elastic. But when you stretch it, it goes through a phase change, and it goes from a solid to a liquid, and then it slowly resolidifies. That's called the phenomena of fluidization. So when you took that deep breath, you fluidized every adherent cell in your lung, and especially the airway smooth muscle. I'm actually, my main area is asthma and bronchospasm, and we're interested in those smooth muscle cells because they wrap around the airway and they constrict like a boa constrictor when they're activated. But in normal people, even if you activate the muscle, when you take a deep breath, you fluidize the muscle, all the contractile force goes away. So who cares about deep inspirations anyway? Well, she does. So this is a classic picture of a girl having an asthmatic attack. And she looks like she's going to die because she is going to die unless someone intervenes. And the reason is, now I already mentioned, this is the airway. This is a small airway inside your lung. It's surrounded with alveoli, and that's smooth muscle that wraps around the airway. And in an asthmatic, someone who dies of asthma, instead of looking like this with a nice big tube that you can breathe through, there's more smooth muscle, there's thickening of all these compartments, there's a mucus plug, and you can't breathe through that. That's why asthma is a bad disease. And as I already told you, with deep breaths, airway smooth muscle fluidizes. And it turns out that just taking a deep breath, it's the most potent bronchodilator we know of. It totally relaxes airway smooth muscle. And you take deep breaths. You've never probably counted, but you take a deep breath about once every 6 minutes. You don't realize, but you do a sigh. So that's relaxing the airway smooth muscle. But in asthma, it turns out the mechanism fails. And some people had even thought that this is the proximal cause of why asthmatics get into trouble, because the most potent bronchodilator that we carry around with us all the time doesn't work anymore. So that was the problem that I'd been working on for most of the 1990s and early part of this decade. And it got me interested in, as we started to get more and more reductionist and trying to think about mechanical properties of cells, that's how we started to get into the rheology of cells. So one of the themes that I want to talk about is that the cytoskeleton, not only is it elastic, but it's fragile. It's made of elastic bonds, but they're all weak. And every time you stretch it, you can just pull the system apart. It's a very fragile system. I'll show you some of the evidence for this in a bit. So just to give you kind of a little bit of an overview of what I'm going to talk about in terms of the mechanics. So biologists like to think about the cytoskeleton as a platform for signaling events, because there's lots of signaling events that go on or are generated by the cytoskeleton, but it's also a physical material. I mean, I'm a mechanical engineer by training. I don't know much about signaling, but I know a little bit about mechanics. So this is a structure. It can deform, it can contract, and I'm going to talk about how you image forces in cells. And it also remodels. It's not a static structure, but cytoskeleton is something that's turning over very, very rapidly. The bonds are always turning over. It's a very dynamic system, and it's burning fuel, ATP, all the time. So one of the big questions, and the ones that we focused on a lot, is what are the physical laws that govern the cytoskeleton? If you think about it from the point of view of material science or physics, well, what are the laws? We know about Hooke's laws for elasticity. We know about Newton's laws for viscosity. How do cells behave? First of all, they're fragile, and it turns out this fragility turns out to be universal. Every adherent cell type, and actually most circulating cells, have the same kind of generic behaviors, which are very, very strange and don't fit what you read. If you're any of you material scientists, mechanical engineers, you all know about viscoelasticity. Forget about it. It really doesn't work in cells, and I'll show you why. And if we can understand these laws, it's important because it'll help us understand how biological systems behave, not just in asthma, but migration, that's what I'm going to talk about a little later, wound healing, barrier function, invasion, epithelial mesenchymal transition, you're going to hear about that tomorrow. Development, we just heard about, and actually, if we have a little time at the end, I can talk a little bit about evolution, how this fits into an evolutionary perspective. But I'm going to emphasize mechanics, materials, and physics, not signaling. Okay, so I've structured this hour as kind of like a play in 4 acts. So one is about this fragility, cells as fragile objects, trekking the rugged landscape, that's collective cell migration. And here I'm going to talk about, if we get to it, and it's not important that we do, but if there's time, evolution, how primitive cells are. We're made out of actually very primitive material, and the question of the last common ancestor. So does anyone know who this is? Anyone recognize that person? Okay, here's a hint. He was at Harvard Medical School in the first half of the previous century. Okay, not good enough. Okay, how about, if any of you heard the word homeostasis? Who's heard of homeostasis? Okay, this is the guy who coined that word and that idea, homeostasis. That was an idea, it was so important that even people outside of MIT recognized it. That was a joke. Okay, this guy is Walter B. Cannon. So he's the guy who came up with this idea of homeostasis. It's everywhere in biology. In his book, Wisdom of the Body. And what he said was this. He said, when a factor is known which can shift a homeostatic state, it is in one direction, it's reasonable to look for factor or factors having an opposing effect. So the whole idea is that in biology, things are regulated. There are positive and negative things that somehow lead to a balance and stasis, homeostasis. Okay, so I'm going to use that idea to talk about the fragile objects. So this happens to be a rat pulmonary vascular endothelial cell. But what I'm going to show you turns out to apply to all cell types. So I didn't mean to advance that just yet. So this is sitting in a culture dish. And it's not just sitting on the dish. You all know there's adhesion molecules. It's adherent to the dish. And it's not just adherent. It actually, when a cell becomes adherent to a dish, it pulls. It makes contractile forces. So this cell is actually pulling on that Petri dish. And it turns out, methods I'm going to tell you about, you can actually image those forces. So this is what's called a traction map. So these forces, as the cell pulls on the substrate, those are called traction forces, and there's a technique called traction microscopy where you can actually measure the distribution of forces that the cell is exerting on the substrate. So you can see there's hot spots here and here tend to be at the cell ends when the cell gets polarized like this. And if you can't make out the arrows, they're pulling towards the center. So this cell is pulling against its substrate. It's contracting against the substrate. That's a universal property or nearly universal property of all cells. So that's true. Every adherent cell is in a state of global tension. And what happens with a mechanical challenge like stretch? So stretch happens all the time. Every time you take a breath, every time your heart beats, peristalsis in the gut. During development, you see cells were being stretched. And I know in development of the lung, in the embryo, there's actually waves of peristalsis that go down the airway, which is thought to be crucial for development. So stretch is really, really important. So what happens to these forces when you stretch the cell in a physiologic way? So there's two ideas in the literature. Okay, one of them is called reinforcement, and that was discovered by Michael Sheets, who's you're going to hear from in this course, right? He's one of the leaders, maybe the leader in mechanobiology. So he discovered this phenomenon called reinforcement. That was his paper. Many of these are his, not all of them. And the notion of reinforcement is if you attach a microbead to the cell, so you coat it with a ligand, let's say for integrins, and you use optical tweezers or laser tweezers or magnetic tweezers, you can pull on the cell, you can tug on the cell. And it turns out the cell, it has passive mechanical properties, but it's also an active system, and the cell responds actively to the force. And when you stretch it locally, it turns out the cell pulls back. So it makes an increased local force. It increases its local stiffness, and it actually builds up structure. So if you look here, you'll see more focal adhesion proteins, more actin, because the cell has detected that, and it responds actively. And in Sheets' paper, he talks about this as being a mechanoprotective mechanism. So the cell detects a force, and it reinforces itself, right? That's a logical mechanism. So he called it mechanoprotection. The interesting thing is, it's totally the opposite of what I've been telling you about with fluidization. These are papers from my lab where we found when you stretch a cell, it doesn't stiffen. It turns to mush. So which one is it? And when? And neither of these is wrong, right? But it's totally a response of the opposite sign. So what's going on? So Ramakrishnan had been a fellow in my lab. He's now got his own lab at one of the hospitals across the river. And he was able to develop a technique where he was able to combine traction microscopy with stretch. So you could do both at the same time. It's a very simple technique. I'm not going to describe it, but it's in the literature. It's so easy to do, it's almost embarrassing. That's why it's such a great technique. So here, this happens to be an airway smooth muscle cell, and that's the static traction map. Here's I think that's phase contrast image of the same cell. And so what he did was he put this cell through a stretch just like you did earlier. So this is cell strain time, and so here he took a 4-second stretch of a 10% uniform biaxial stretch. So that's right in the middle of the physiologic range as regards lung. And so the question is, what happens to these contractile forces? Right? If there's reinforcement, they should get bigger. And if it's fluidization, they should get smaller. So what happens to the traction response? So here's the movie. Maybe can I do it from here? So So what you're going to see when I run this movie is you're going to see a few seconds of baseline. You won't see what happens during the stretch, but then you'll see what happens after the stretch. And up here, this is actually a graph. You can integrate these contractile forces and quantify them, and you'll see here what happens to the contractile forces. So here's the movie. Is baseline. Stretch happened right there. As you can see, the contractile forces just got totally wiped out. I mean, this is 0 to full scale. They went virtually down to 0, at least over the time scale we could. I'll play that again. Baseline, stretch. So as fast as we can measure, the forces go to 0. Basically, the cytoskeleton got ripped to shreds, and actually, if you look, the actin filaments disappear, actually. We don't understand how that happens, but they disappear, and then they slowly reassemble. I don't know what happened there. And the graph is gone. And then over the course of about 600 seconds, you get a recovery, which is about the time between deep inspirations in healthy people. So that's the fluidization response. Oh, sorry. Well, you're going to see it again. And there's the graph showing the recovery over about 600 seconds. Okay. So I'm not going to show you all the data, but it's a fluidization response. The cytoskeleton went from a solid-like to a fluid-like state. And it turns out this seems to be universal. We've looked at about a dozen or 15 different cell types. It seems to be a universal phenomenon. So coming back to this question, so what's going on here? Well, and why do you get different answers? Well, we don't know, but our speculation is this. In this case, all these investigators looked at a kind of a local response, pulling on a microbead. And what we had originally been thinking is that and I think what these people were thinking is, if you know what's happening locally, then it's easy to predict what's going to happen globally. You just add it all up. For those of you who, if you know the Green's function, if you're into differential equations, you can just add it all up. So that the notion is, if you understand that response, well, you can apply a load everywhere homogeneously and simultaneously, and the global response should be the sum of the local responses. That's what we had been thinking. It turns out it's wrong. And it turns out it predicts responses of the wrong sign. It's not a matter you're off by a factor of 2, you get the wrong direction of the response. So that makes it interesting. And we speculate, we don't know this for sure, but we have some evidence that suggests it has to do with gradients of strain. When you homogeneously stretch a cell, which pretty much is what you do when you're breathing or your heart's beating, you get one kind of response. But when you have a localized load, you can get big strain gradients. And that seems to give a response of the other sign. So at least in terms of physiologic types of stretches, we say the fluidization trumps reinforcement. So you can ask Sheets if he agrees with this. He may or he may not, I'm not sure. So we think that that's the physiologic response. It could be that reinforcement is some kind of an injury response or something that, in any event, it's a much more local response. So just to summarize what I've told you so far, the cytoskeleton is fragile, and this is a big effect. This is not like an adjustment to some kind of other idea. It's a dominant effect. It's not secondary. And in terms of the way biologists tend to think about mechanotransduction, usually we think about either a chemical force, that is ligation of some receptor, or a physical force as representing a first message. Then it's somehow transduced. Then there's a second message. Then can be a very complicated signaling cascade and an end effect. And a lot of the literature about mechanotransduction thinks about it this way, that everything is always mediated by such a pathway, which is, of course, much more complicated than I've sketched it. And actually, here's a recent paper looking at one of the important pathways. But in addition, the cytoskeleton's a material, and it's a fragile material, and it can be direct effects of physical force on the material that aren't necessarily mediated by signaling. It's just a material that you can pull apart. And if you can pull it apart, then it's going to impact all those other steps in the process. And of course, there can be end effects that are mediated by signaling. They're going to change the material. So it becomes a really rich and interesting problem to study if you're approaching it from the point of view of physical science. So is the cytoskeleton fragile or resilient? That's an interesting, we find it pulls to pieces quickly and easily. I haven't shown you what happens to the molecular mobility, but it turns out it goes up by 2 orders of magnitude. When you stretch it, that's changing from a solid to a fluid. And the reassembly, I haven't shown you, it's dependent on ATP and certain tyrosine kinases. So this is another mechanoprotective mechanism, right? So one is to reinforce, right? If you feel the physical force, you reinforce. But another is fluidize, just go with the flow. That's another way to prevent injury. So it's another mechanism of mechanoprotection. So let's talk a little bit more about material properties of cells. So this happens to be an airway smooth muscle cell, and that's not the nucleus. That's a magnetic microbead that we coat with RGD that binds to integrins, and we use that as a mechanical probe. So let me just ask all of you, if you imagine that you can, right, given that you're here, you're a very self-selected group. You've been thinking about cell mechanical properties at least a little bit. So imagine that you could shrink yourself down to the scale of a cell, and you could poke it with your finger. What would it feel like? Have you ever thought about that? Anyone want to make a suggestion of what it would, if you could imagine you shrink yourself down, just poke it. What does it feel like?
It should be soft.
Soft. Good. How soft? Can you think of a material that you'd say was comparable to?
Maybe like a waterbed.
A waterbed. Good. Good. You're off by 4 orders of magnitude, but that's good. That's good. You'll see why in a bit. Anyone else?
What mayonnaise?
Yeah, well, you've probably heard me talk before. Yeah, it's about like mayonnaise or toothpaste or shaving foam, right? Y'all, all of you have looked at Gillette Foamy. That's roughly how all those things are on the order of 1 kilopascal Young's modulus. They're really, really soft. And it turns out that's roughly where cells are. And they're roughly of the same consistency, right? I could take, if I had a bottle of Gillette Foamy, I can make a big pile of foam. It's actually not going to flow, right? It'll just sit there. It's an elastic body. It's a weak elastic body, but it's elastic. If I put a shear force on it, I can fluidize it. But it's actually elastic. That's roughly the mechanical behavior of cells is comparable to Gillette Foamy or mayonnaise or toothpaste. So that's where we're at. So actually, that's shown here. So here's Young's modulus. That's 11 orders of magnitude. Right? That's how we think about elastic property. A diamond is up here. It's one of there are actually things that are stiffer than diamond, but a diamond is usually, in nature, that's the stiffest thing you find. Here's all engineering materials. Here's some biological material. Bone is really stiff, 10 to the 11th pascals. Actin filaments, really stiff, 10 to the 10th pascals. Elastin is like rubber. Here's a foam polymer. Waterbed, the foam polymer is about 10 to the 6th, 10 to the 7th megapascal. Bacteria, I probably won't get to the evolution part, but bacteria are here, they're about 10 to the 6th. Eukaryotic cells are down here. They're 3 to 4 orders of magnitude softer. These things are really, really soft. And they're comparable to foams, paste, colloid suspensions, cold cream is another one. Ketchup, that's where eukaryotic cells are. There's been a lot of work in the biophysics literature trying to model a cytoskeleton of eukaryotic cells. It turns out actin gels are actually about 2 orders of magnitude softer. So it's really hard to get an actin gel in the lab that will behave like a cell unless you actually tense it. It turns out tension turns out to be a crucial part. You can mimic eukaryotic cells with an actin gel if you shear it or stretch it. Okay, so let me move on. Let's see. When should I stop talking? I've got
a full hour.
Okay.
And then a half-hour discussion after that.
Okay. So let me I'll say a few words about this. I wasn't sure how much time I should spend. So I've already told you that the cells they're elastic, they're very soft, and it turns out if you look at this structure, you'd like to be able to predict the stiffness from the constituents, right? Here's the actin filament density, here's the cross-linking density, alpha-actin, and so on. And if you try to do that, you actually can't predict the stiffness very well. And the reason is that the stiffness of this system depends on the tension. And you can't really see the tension here. But if you know the tension, you can actually predict the stiffness. And that comes from these papers. So tension turns out to be crucially important in the whole story. It's just not a matter of passive mechanical properties of the molecules. The rheology is weird. Okay, so I said viscoelasticity. I don't like viscoelasticity. And the reason is this. When you look in the textbooks about cell mechanics, they'll all say, well, let's postulate some kind of viscoelastic model. And some of you know what those are. It turns out all those models have built into them certain time scales, or viscoelastic time constants. It turns out when you actually measure the mechanics of this system and you do it over a very wide range of frequencies, like 4 or 5 or 6 orders of magnitude, you can't find any time constants. What you find is that the elastic modulus goes, omega's frequency, it goes like frequency to some power. It's a power law. And alpha is typically about 0.2, 0.1 or 0.2, so it's a weak power law. And just if you make a graph, you just see stiffness progressively increases as you go decade after decade after decade of frequency. The stiffness just keeps going up as a straight line on log-log paper. So you can say, well, okay, I can model that with an infinite distribution of time constants, but it turns out that that's not a satisfying, yeah, you can model the data, but it's not mechanistic. And the interesting thing is things like pastes and foams and slurries, physicists, soft matter physicists call those soft glasses, and the rheology of those things just still is not understood. It's not understood. And work from my lab, actually, these papers show that cells belong to that class of soft glassy materials. And this is one of the features of power law rheology that really you can't represent, at least mechanistically, in terms of viscoelastic models, because the stresses really, the viscous stress, the frictional stresses are not viscous. I'll get to that in a second. Also, it turns out the mechanics depend on the rate of ATP utilization, especially the friction depends on the rate of ATP utilization. So it's different than other, it's even other soft glasses in that regard. So at the molecular level in the cytoskeleton, frictional stress is not a viscous stress. So those of you who know about viscosity, the idea is you shear a fluid and there's momentum exchange as molecules go back and forth from higher to lower velocities. That's probably not what's going on in the cell. It's probably more like there are bonds that hold it together. And when you stretch the system, the bonds store elastic energy, and then when the bonds break, that energy gets dissipated, like Velcro, right? Right? You could peel Velcro, and it's going to absorb a lot of energy, but it's not viscous. It's more you're pulling, stretching those springs, and then they go boing, and the energy's gone. So it's not a viscous mechanism. It's more like breaking of bonds that then reassemble in a manner that's dependent on ATP. I've already told you about fluidization. You can look at the nanoscale of motions in this system, and it's really interesting. I think you mentioned something about thermal agitation, right? So there is thermal agitation here, which is making molecules move. But it turns out much bigger than that are the motions that are driven by ATP hydrolysis, because ATP releases about 20 kT. So kT is thermal agitation. Hydrolysis of ATP is 20 kT. And it turns out if you look at the motions here, the Stokes-Einstein relation. Any of you know what Stokes-Einstein is? Some of you. Okay, so Einstein in 1905 said that basically viscosity and diffusion are the same process. If you know diffusion, you can predict viscosity and vice versa, because it has to do with molecular collisions and thermal agitation. Well, it turns out it doesn't work in cells. It doesn't even come close. So that breaks down. And the motions that you find are molecules get caged and then they hop. They get caged and hopped. So the rheology is just, it doesn't fit. By the way, viscoelasticity would say that Stokes-Einstein has to hold. Collective cell migration I'm going to talk about in a second, and let me not say anything about that. So just to recap, we can think about cytoskeleton as a spatial template for mechanotransduction downstream signaling, or we can think about it as a material for stress transmission that can contract, deform, and remodel. So this is just to say, this is an important paper by Chan and Odie that appeared a few years ago where they looked at how cells spread and move. And the notion is that there are bonds that form and break at the leading edge that are related to fragility, so they related to motility. So the system has these on and off springs that can dissipate energy. This is a different model. This is called the glassy worm-like chain, but it has some of the same features. It says that actin filaments can get stuck on one another. And it turns out this model actually maybe is the best one for understanding cytoskeleton. It's a very nice model. What these have in common is that they say the system is weak, it's flimsy, it's fragile, you can pull it apart, it fluidizes, and in this case, you can say that these bonds act like some kind of a clutch that you can pull apart. This is the traditional viscoelastic picture, by contrast. This is from a paper by Bausch over 10 years ago, which had been the classic of cell mechanics. This had been the paradigm. Some of you know about the tensegrity model from Ingber. Well, if you listen to Ingber talk about this, he emphasizes that this structure is stable, robust, resilient. Well, it is, that is, but I think cells probably this captures some features of cells, but certainly not all of them. Okay. So let me move on to collective cell migration. So this is a monolayer of MDCK cells, Med and Darby canine kidney cells. And let me just play the movie. It's a lot like the one you already saw, but a little different. So this is like a wound healing assay. So there's no cells here. Here are these cells, and they're moving to fill the wound. Okay. Things like this have been measured for a long time. Oops, sorry. Let that play. Okay. Oops. Okay. So those are cell motions. The earliest paper I know of is 1914, where motions like these were analyzed. The question is, and the question that my lab became interested in is, what are the physical forces that are associated with these kinds of motions? Can you relate the forces to the motions? It's actually not obvious that you can when you think about it. Maybe I'll get into this in a second. And when you look at this, it looks like the cells are flowing, right? And in some sense they are flowing, but are they behaving like a liquid? Or are they actually more like solid-like? Actually, I'm going to show you they're actually, even though they're flowing, they're solid-like. So how can you understand this problem of collective cell migration? And actually, let me interrupt myself by saying the problem of single-cell migration has been a big problem in the last 20 to 30 years, and it's now pretty much worked out. The problem of how a single cell in isolation crawls, now there are really good models for that. Collective cell migration is a totally different story. We don't understand it. So how do you understand these kinds of motions? Well, it depends on what literature you read. So one area of the literature, it says that it's all about leader cells, that there are specialized cells at the leading edge. They've been studied a lot, especially recently. You can see this one, this one, this one. And the notion is that these are cells that are specialized for pulling. They're like the locomotive of a train, and they're pulling the sheet. So these are the ones that are pulling, and they're pulling the sheet to the left. So if the model would be, imagine you're in bed and you grab the edge of the blanket, right, and you pull it up under your chin. So all the motive forces at the edge, and you pull the sheet along. So the implication there is if it's all about these leader cells, that means the cells behind must be in tension. Okay. So that's the model that really dominates injury and wound healing literature. Okay. Here's another idea, and that's that each individual cell is mechanically self-propelled. Right? Each cell has its own motive force, potentially. And this would be like soldiers marching in a column, right? All the soldiers are marching along together, but there's no force of interaction between them. Each one is self-propelled, or cars in traffic. So in that case, the stress in the layer would be close to zero. So there's a whole literature on this kind of either modeling cells as being diffusive motions or fancier models, Levy flights, things of that sort. In the cancer and development literature, you find more about the notion that there's proliferation behind the leading edge, and that proliferation actually pushes the front forward. It expands the colony and pushes the front forward. So the notion there is that actually the cells here would be in compression, because you get more and more of them with time, and it pushes the front forward. So actually, I'm just curious, how many think it's this one? Good, okay, and this one? Nobody? This one? Okay, and so the rest of you don't think anything, right?
But should they be like exclusive? You sure?
Not necessarily, not necessarily. And it could be in different, it could be one in one kind of system and one in another. Could be. Like, for example, in tumors, I mean, people have measured pressure in tumors, and it's compressive. But in a monolayer like this, even there, you know, people argue about it. And the problem is that there had been no way to actually measure the stresses in the monolayer. How do you measure the stresses? So it's been, you know, people have looked at things like, look at stress fibers, and the cell looks like it's being pulled this way or pushed that way. But it would be really nice if you could actually measure the stresses. Well, they had been inaccessible until recently, but it turns out you can now there are techniques to make the forces visible. So let me show you some of them. So here's this same monolayer, and it's just all MDCK cells, nothing fancy about it. So first, it turns out this guy, Javi Tripat, had been a fellow in my lab. He's now a professor in Barcelona. He took this traction microscopy that I already showed you, one traction map for a single cell. He and Jim Butler figured out how to do the same kind of thing for a whole monolayer. And it turns out it required some doing, but now it's done. So no one has to redo it. The math is done. It turns out to be not easy, but they did it. And they measured the traction forces that the cells are exerting on the substrate. Okay, so when you look at that, this is the picture that you see. So let me orient you. So again, these are the forces, or the shear stresses that the cells are exerting on their substrate. The warm colors are cells that are pulling themselves to the left. Okay, and the blue here is actually cells that are pulling the other way. And when we first saw this, we were very, very surprised for a couple of reasons. One is it's heterogeneous, right? It's punctate. It's not a smooth picture. We thought whatever we'd see, it would be smooth. It turns out it's not smooth. Cells that are nearby each other pulling in different directions. You can see it's mostly red at the leading edge. So those are the leader cells, and they are uniformly pulling to the left. But it turns out when you add up the forces, it turns out there's a net, in general, cells are pulling to the left, and these actually add up to much more than what's happening at the front edge. You can actually play the movie. So not only is it heterogeneous, here's the movie. The stresses flicker on and off. So it's not like one cell is always pulling in the same direction. It'll be pulling in one direction at one time, and another direction and another time. So it's not the position in the gel or the particular cell. It's something that's dominated by fluctuations, which is really interesting. Oops.
What is the resolution of your force measurement?
It's about what you can see here, which is a pixel. So it's about 5 microns, something like that.
A few pixels per cell, right?
No. 1 pixel per cell, you said? No. No, so here, these are individual cells which maybe are 10 pixels across, something like that. And these are like, I can't remember how big the pixel size is.
So what's the distance from the leading edge shear cell resistance?
This is, I don't remember. I think 100. I lost the scale bar, which used to be here. Maybe that's 100 microns, something like that. Yeah.
How would the stress distribution change if you begin imaging right from the start where you have a free edge? So
I'm going to show you that. That's a wonderful question. It's a paper, actually, Roger might have sent it to you, I hope. The paper just appeared on Sunday that answers that question, and I'm going to try to get to it.
I'll post it if you send it to me.
Oh, okay, it's posted, okay. But that's a wonderful question, and I'll show you the answer in a bit. There we go. We can look at the same data a different way. So here's a piece of bare gel, and we can look at the traction force at that point as the cell monolayer sweeps over it. So here's traction. That's the noise floor, and here come the cells. Every time I see this slide, I'm amazed. So what you see is there's nothing, and then actually the first lamellipodium, there's actually a little bit of pushing, but then it's these huge fluctuations. And what we had thought, we thought we were going to see some smooth kind of signal with maybe little fluctuations, but it's not. It's huge fluctuations with a small baseline. So there's a net tension. The monolayer is basically in tension, but that mean value is swamped by the fluctuations. So it's a very heterogeneous system. So if one is interested in mechanotransduction and how certain kind of binding or signaling is force dependent, the idea that you're just going to use some kind of a mean field theory, average it all out, that it's smooth, it probably is way off.
It's very interesting. So is there any characteristic time scale of the oscillation, or is it like power law distribution?
Yes, there is, and it has to do, we've got another paper I'm not going to talk about. We have reason to think that these cells are close to what's called a jamming transition, like coffee beans that get jammed in a chute. And it turns out as the cell density goes, I'm not going to show the data, but as cell density goes up, the fluctuations get spatially bigger and slower. And you can get usually packs of cells from 5 to 30 packs all moving together. That depends on a number of things, including cell density. So it depends, and you need a different kind of frame to think about it. And we think jamming or traffic is something that's related to that. But I'm not going to talk about it. But we published a couple of papers on that. Okay. Oops. Sorry. Okay, so it's all about the fluctuations, and they're severe, and it turns out most of the action is far behind the leader. It's not about the leader cells. I think leader cells, okay, that's another paradigm that or dogma that leader cells are interesting, and they maybe give guidance, but in terms of pulling the monolayer forward, they don't actually do much. Now, this is interesting. So here's that circle and that one are the same. So that's a pack of about 2 or 3 cells, 4 cells maybe. And these guys, they're moving to the left. But in terms of the forces they're exerting on the substrate, it's as if they're pulling in the other way. So either they're trying to go against the stream, or maybe they're being dragged along and they're dragging their feet. So what's going on here? And why are they moving the way they're moving? Well, in terms of people who study cell motility, the mechanisms that are known are kind of chemotaxis, so cells want to move along some chemical gradient. Durotaxis, cells can feel differences in elastic properties of the substrate and usually want to go from softer to stiffer. Haptotaxis is there can be differences of adhesion, and cells might want to go up an adhesion gradient. Well, it turns out none of those apply in this model system, so something else is going on here. Somehow cells are swept along by the crowd. And I'm going to get into this. It's called now we call it Plutotaxes. So now to understand that, you have to understand that this is only part of the mechanical problem. Right? So these are the forces that the cells exert on the substrate. But what about the forces they exert on each other? Right? That's a different matter. So this is where Donna J. Tombe and Corey Harden in my lab actually figured out a way to compute those. So here's the physical picture. Here's a monolayer, and these red arrows represent the traction forces here. So those are the forces that the cells are exerting on the substrate. And it turns out it can be like a tug of war, right? So here these men are pulling on a rope, and it turns out if you know the traction forces, it's really easy to figure out the tension in the rope. Right? The tension in the rope is 0 here. It's 1 unit here, 2 units here, 3 units here, because Newton's law says you have to have a force equilibrium, right? Is that clear to everybody? Obvious? So what that means is if you know the traction forces, you can actually then compute the intracellular forces, the forces that one cell exerts on the other. It's almost as simple as that. So here you can put that in terms of mathematics. So here's the monolayer. Here's how we actually do the experiment. There are fluorescent microbeads here in the gel. This is a soft gel. It's about 1 kilopascal. You can measure the deformation of these particles, and that gives you the traction force. That was something that was worked out by Trepot and Butler. We put other beads here to correct for stage drift. And then if you know the tractions, you can just add up the forces, because the force balance is the stress here minus the stress here has to be the traction force. So from the measured bead displacements, you can compute analytically the traction force. And you can do that pretty rigorously. And then once you know the traction forces, this is just Newton's law. The divergence of the stress has to be the applied force. And so you can get this internal stress field. And it turns out it's almost model-free. So you say, do you need a model to do this? Well, sort of, but the assumptions are really weak and not really a problem. So one last thing before I show you the result. So if you can then compute the state of stress everywhere inside the monolayer, so this is the typical engineering way of representing stresses. So that's the sigma on the x face in the x direction, on the y face in the y direction, shear stress on the y face in the x direction, shear stress on the x face in the y direction. So you all maybe know, or if you don't, you do now, that you can rotate your laboratory frame, and you can, if you find just the right orientation, these stresses can always be converted into these. This is called the maximum principal stress, the minimum principal stress, and the principal orientations, like this. And the nice thing about this description is when you find this orientation, shear stresses are zero. So there's zero shear stress on those faces. So this state of stress and that state of stress are exactly the same. It's just a mathematical trick. It comes from Moore's law, for those of you who know what that is. So this is totally equivalent. So that's nice. So you can find orientations where the shear stress is zero, faces where the shear stress is zero. So let's look at the monolayer. So now I'm switching. This happens to be endothelial cells, but everything I'm going to say applies to epithelial cells as well. What happens if you look at the tensile stress? This is now the cell-cell forces. Again, we got a bit of a surprise. So this is a kind of a relief map showing, if you will, cytoskeletal tension or tension between cells at cell-cell junctions. This is 0 up to 300, and what you find is you find these kind of massive peaks. So that peak there comes from a cluster of about 50 cells here. And here's another kind of mountain range here, which corresponds to those. And these are a bunch of cells that are working cooperatively to build up tension, passing it from cell to cell. So you get a rugged stress landscape. So this is rugged, and so as the cells migrate, they're maneuvering in a stress landscape that's very, very rugged. They're also creating that landscape because of this cooperative stress pileup, this notion that one cell pulls on its neighbor, pulls on its neighbor, pulls on its neighbor to make structures like those. So there's a tension pileup, and the stress landscape is severe. So this is just tension. So this is kind of averaging over all directions. So some of you more mechanical guys might ask, is the stress isotropic? And it turns out it's not. So here, this is actually it's complicated but interesting. So each one of these ellipses, take that one, represents the state of stress at that point. And that's the principal orientation. The maximum principal orientation this way, the minimum principal orientation is that way. So you can see the stress is not isotropic, right? The tension is higher going this way than that way. And you can see that there's correlation, that these ellipses tend to be organized in swirling patterns that are coherent over many, many cell bodies, over like 50 cell bodies, something like that. So you see this swirling So the stresses are distributed in this interesting heterogeneous pattern, but there's lots of correlation across cells. It's not totally random. So that's the stress landscape. Now what I haven't told you yet is what the red arrows are. The red arrows are the instantaneous velocity of the cells, the migration velocity. And when you just look at this, what your eye seems to tell you is that the cellular velocities tend to be aligned with the maximum principal orientation. So like this and like this. And it turns out that that's exactly right. And I think I might have some data I'm going to show you. So it turns out, and this is, I think, an important discovery, that cells tend to move along the direction of maximum principal orientation. And that's something we hadn't known before. And there's another very interesting implication that isn't worked out yet. So here's a cell. And here, let's look at that cell-cell junction, and here's the state of stress at that cell-cell junction. There's normal stresses, tensile stresses, and a shear stress. Well, this says that cells are moving along the maximum principal orientation. And I told you, based on Moore's law before, that along that orientation, the shear stress is 0. So what this is saying is that cells tend to move. They're happy to push and pull on their neighbors, but they can't support any shear stress. So that's actually very interesting. So along the principal orientation, the shear stress is 0. So cells tend to trek along shear-free trajectories. So these motions, the cells are minimizing the shear stress between them and their neighbors. They're happy to push or pull, but in terms of if they shear, they can't support or don't support shear stress. So this phenomenon, it's not chemotaxis, it's not haptotaxis, it's not durataxis. Instead, it's some innately cooperative and collective behavior. So we gave it a name. We call it Plutotaxes. So from the Greek word plithos, from a crowd, swarm, or throng. And the principle is that neighboring cells join forces to transmit normal stresses across cell-cell junctions, normal meaning perpendicular, but they migrate along orientations of minimal intercellular shear stress. So what that says is either the adhesion molecules here, let's say either tight junctions or adherens junctions, well, there'd be no tight junctions, or few tight junctions here, because this is endothelial. But it's saying that those cell-cell adhesion molecules, either they're not able to support shear stress, or somehow they remodel so that they don't support shear stress. And we don't know which one it is. So, and we know that this is true of epithelial and endothelial monolayers, and even breast cancer cell lines before the EMT, but not after the EMT. So that's Plutotaxes. Let me just move on. So this is just a blow-up of that same picture. Let me just skip. This is actually the doc. I'm going to skip this. This is just showing that the cell motion is highly correlated with the maximum principal orientation. So that's the statistical support for.
Jeff, when you say maximum principal orientation, you mean the actual orientation, not the maximum principal stresses?
I mean the orientation. So in the stress field, there's a maximum and a minimum principal stress orientation.
Okay.
Yeah,
So
stress.
Not the elongation of the cell.
No.
Okay.
It turns out in cells that are elongated, they also tend to elongate along the maximum principal stress orientation. That's for endothelial cells. Epithelial cells are more like fried eggs and don't have preferred orientation. Yet, even though the cells aren't oriented, the motions and the stresses are oriented.
Excuse me. So, in order to determine the principal directions, you have to know the shear stresses.
I'm sorry, could you say that again?
To know the principal directions, first you have to measure the shear stress.
Yes, that's right.
So that's
We measure, yeah, actually, from that, I went over it fast, but from the force balance in the monolayer, we can get all the stresses inside the monolayer, the normal stresses and the shear stress. I didn't show you the maps in the interest of time, but yeah, we have those numbers are there, and they can be pulled out and mapped.
So I noticed in some cases in most cases they line up in the direction of motion.
Not always.
Yeah, but in some cases.
Yeah, actually, let me go back. That's good. Yeah, so So it's a statistical thing. So here, you get very good alignment, yet let's say here it's not so good, here it's not so good. So it turns out that where the stresses are the most anisotropic, that is when these ellipses are more like spindles, that's when you get the strongest relationship. When there's no preferred orientation, let's say, are any of these like circles? Well, maybe that one, this one. So if there's no preferred orientation of the stress, you can't find a correlation between the motion and the principal orientation.
If you look at the bottom right,
Here?
yeah, there's a spindle one.
Which?
Yeah.
Tell me.
Yeah.
Here?
Right. Yeah. Right.
Yeah. Well, yeah. It's bad. Oh, here?
Yeah.
Yeah. Yeah. The correlation is bad. So it's a tendency. It's a tendency. Yeah.
So are they always intentional?
Almost all. You can find, if I go back, you can find pockets of compression. They tend to be small and few. Mostly it's overwhelmingly tensile. Overwhelmingly tensile, although you can find regions of compression. Yeah.
I got the point that the motion is injection of the maximum stress, but I'm having a hard time to connect this to the collaborative behavior.
To the collective behavior. Good. Well, me too.
And how do you reason that this is due to?
Well, I mean, the stress field itself is somehow cooperative, right? Because this correlation that you see, and we've quantified in other papers, the cells are not behaving independently. It seems to be all about the cooperativity and how one is pulling on its neighbors. So it's some kind of a cooperative phenomenon. And similarly, the velocities are hardly correlated. And here's actually a deep question that I don't know how to think about. You'd like to think that the forces steer the motions, right? That's, as engineers, we like to think about that way. The forces steer the motions. But is that what's happening? Maybe. Another could be cells just decide I'm going this way. And it could be the stresses are just a byproduct of the motion, right? Because they're mechanical objects, they have certain elastic properties, and they go where they go, and there's going to be it's going to cause stresses. So put it another way, what's the cause and what's the effect? And actually, we don't know. We don't know.
So does the idea F equals ma apply here?
I'm sorry.
The idea of F equals ma apply here?
Not well, okay. The inertial no, yes and no. The notion that there's a force balance, and that maybe there's an equation of motion, like right. If we were fluid mechanics guys, like me and Roger are, you'd say, well, if this was a fluid, we'd say it's Navier-Stokes, which is just a force balance of each little element here. I said no because inertia there's no m in this problem, and the accelerations are small. So there's no inertial effects, but there's elastic stresses, there are frictional stresses, and there are contractile stresses. And in each element, each small element, those forces have to be in balance. And if you could discover the law that relates the forces to the motions, you have a science paper. Right? And we don't, we're actually, Jay Kim is back there in my lab. That's what he's trying to do. Is what is the relationship of forces to motion? Is there a relationship? It's not clear that there is. It's really not clear that there is. I mean, in fluids there is. In elastic bodies there is. In cells it's not. It's actually not clear. There's an old saying in physics that it's hard to find a black cat in a dark room. Think about that. Hard, especially if there is no black cat. That's also, it's not exactly a joke, but it's supposed to be funny. Yeah.
Is it that the cell chooses the direction first and then the stresses conform?
Yeah.
It might be that the ones that don't line up, maybe the cell has just chosen the direction in its brain, or mathematician's brain.
Uh-huh.
And then it's about to do its physical change, but you've just taken the shot before it's made the change, and so that's why they don't line up. We thought about
I don't think I understand. I don't understand what you said. So could you say it again? I didn't follow.
So if the cell chooses its direction that it wants to go in,
Yeah.
and then it makes its body shape changes.
Yeah. Yeah.
And so then its stress circle changes.
Mm-hmm.
So it can move. What if there's had to be a delay between the cell deciding to go one way and making the change? So what if you take the photo?
Good. I think, hold that question, because I think it's related to the question that, what's your name? You asked me about the...
Manchu.
Yeah, Manchu. I'm going to show some data that may bear on that. What happened? Oh, there it is. Okay. Okay. So let's move on, and I hope I'll get to something that will help to answer this. Okay. I'm still okay on time?
Well, we would be entering into a half-hour discussion.
No.
So, but there is discussion, so I'm happy for
Maybe I'll finish up in 5. I'll just go quickly over this next. I promised this other stuff, so let me just go quickly over this, jump, and then I'll wrap up. Okay. So. Traction force microscopy. So actually the first paper, really important, Cyril Harris, 1980 in Science. He put cells on silicon membranes, and he showed that the cells can wrinkle the membrane. So that was the first demonstration that cells generate contractile force. Then, I'm sorry, I'm going ahead of myself. Then Micah Dembo over just across the river at Boston University was the first to show that you can actually quantify that and make these traction maps. There was actually Jim Butler in my group who actually showed that you can tremendously simplify the problem, and you can also democratize it. That is to say, Dembo didn't share his software, and no one could duplicate it because it was too complicated, but Butler showed you could do the problem much more simply, and he gave the software away to everybody. So that most people use Butler's technique now. Ramakrishnan, I talked to you about, he showed how you can use the same technique with fluidization and for studying cell polarization. Then in terms of collective cell migration, I've already told you about that. And Plutotaxes. But now, in the last few minutes, let me just talk about the question that this young man asked, which is, what happens when you lift a barrier and let cells go off to the races? So. This is a paper, it just appeared Sunday. So this is a group, the experiments were actually done in Barcelona by Javi Tripat and his students. So here was the idea. They made a polyacrylamide gel, and they put a PDMS membrane on top of it, and they plated cells in this rectangular region. They grew the cells to confluence, and then they lifted off the membrane and to let that rectangle of cells start to expand. Okay, so here's what they found. So here at T equals 0, and this is stably expressed GFP actin. So here's the monolayer. So there's no cells here that have been grown to confluence. And then an hour later, the cells have started to flatten and crawl. And then 4 hours later, they've crawled and flattened quite a bit. Okay. So here's what they find for actin. It turns out the actin cytoskeleton of these cells, you can't really see it here, but you get much more, a lot of transverse stress fibers here that you didn't see here. This is E-cadherin. So these are the cell-cell junctions. And this is really interesting. You see lots of E-cadherin here, but as the cells start to spread, and I'm going to show you that the tensile forces are going up, way up here compared to here, the E-cadherin goes away. This is really, really interesting because there's a number of papers in the recent literature looking at cell-cell pairs, just two cells, and measuring the force at the cell-cell junction and showing that E-cadherin localization goes up with increasing force. But in this system, it goes down with increasing force. So it looks like cell packs are not the same as just two cells kissing one another. This is tight junction proteins. They don't change. This is paxillin, so that's a component of focal adhesion complexes at the cell base. They go up. Okay, so it looks like it's a transition from an epithelial kind of towards a mesenchymal state, but it's not complete because E-cadherin gets partially internalized. It's not increasing with increasing stress, it's going down. And also there's been actually in your field, in developmental biology, Thomas Lecute has been saying that the flow that you find in developmental biology may be going up a cadherin gradient, which would make some sense, actually. But actually, it doesn't. Actually, it's going in the opposite. Cells are going down a cadherin gradient here. So what's going on is actually not at all clear in terms of that. So here's, I didn't download the movies, but this is kind of stop frame. So here's at 15 minutes, 120 minutes, 450 minutes showing this strip of cells spreading. Here are the velocities, the cellular velocities. So initially you see the cells on the right start moving to the right. It's color-coded here. Cells on the left start going to the left, but nothing much happens in the center. The motions start to kind of penetrate from the edges in until you get much later this pattern where cells on the right are mostly going to the right and cells on the left mostly to the left. Here's the traction forces. So these are the forces that the cells are exerting on the substrate. Before you lift the membrane, there's actually almost nothing. As soon as you lift it, you start getting traction forces that appear at the boundaries, like at a boundary layer. So these are pulling to the left, these are pulling to the right, and then with increasing time, they develop more and start, again, there's nothing much in the center, but much later, you see here, cells on the right are mostly pulling to the right, but not totally. Cells on the left are red, they're mostly pulling to the left, but there's still plenty of blue. So again, this heterogeneous picture. And then you can then actually compute the intercellular stresses. This is, I can't remember which component this is. This is average normal stress. This is just cytoskeletal tension. Initially, it's really small. It builds up at the edges and then penetrates in. So this is now the interesting thing. You can take these data and you can simplify them using something called a chymograph. Any of you ever heard of chymographs before? Some of you, okay. So here's what a chymograph is. So here are these data. The problem is it's two-dimensional in space and one-dimensional in time, right? There's X and Y and there's T. But we can reduce the spatial dimensions. I could just take this and average it over Y and just look at the average value of the velocity as a function of X. So for example, at 120 minutes, if I average these, I can then make a chymograph. This is now time versus distance, and this is showing color-coded the velocity distribution at that time here. So that's the color coding here. I could do the same thing for this time, for this one, that's here, and for this one, that's here. So that's the chymograph. And so with this slope, this is time versus distance, that's the speed, that slope is the speed of the advancing front. Same thing here. And what you can see is basically when you lift the membrane and you start the race, nothing happens in the center. And then there's some kind of looks like a wave that penetrates all the way through. And then you get all kinds of interesting phenomenon here. And actually, here you get cells all moving to the right, cells all moving to the left. But here's the reverse. These are cells, there's a pocket of cells here moving to the right. Those are moving to the left. And down the center line, you actually get pulsations. So let me just show you those. So that's the same velocity chymograph. This is the traction force chymograph showing that all the traction forces are very heavily concentrated at the edge, although there is stuff in the middle. And this is the tension chymograph. Again, very little tension at the beginning, and then it builds up high tension in the center here, given enough time. Here, this was the most exciting slide. We looked at the strain rate, so the rate at which cells are expanding, and what happens is you find that there's a wave that's generated when you lift the barrier. a wave gets generated here that propagates across the whole monolayer. So that's a strain rate, a wave that goes over there and it bounces off. And similarly, one that starts here and goes here. And this wave has roughly twice the velocity of the leading front. So this is a mechanism of communication from one edge of the monolayer to the other. And we don't really understand. We have some ideas of what this is. We even have a model in the paper, but I'm not sure that we believe it. So this is a very interesting wave of rarefaction that starts at the leading edge and penetrates the whole monolayer. So they make an X on a chymograph, so we call them X waves. But we don't really know what they are. So again, just to look at these. So here, when you just lift the barrier here, that corresponds to this point. So at that point, the cells are neither moving nor straining. At this point, the wave has penetrated some, but not up to that point. So the cells are not moving or straining. But here, at that point, the cells are pretty much moving en bloc, and they're consolidating. So that's these interesting. I'll stop with this last graph. If you just look in the center here and focus on these cells, the color code here is the average, the stress in the x direction, so that's the tension along this direction. You can measure that tension, and you can also measure cell size here. So if you measure cell area in here, it turns out it's oscillating, goes up and down and up and down, and you can also measure the stress. And the stress goes up and down and up and down, and they seem to be actually in phase. So since the stress and the area are in phase, that means it's an elastic stress. It's not a viscous stress. So there's all kinds of interesting phenomenon here relating forces and velocities and so on. And if someone knows how to figure this out, we'll have a big step forward. The important thing is now we can measure the forces. They're not a matter of speculation anymore. You can measure them and try to drill down a little bit deeper. So I don't know if this answers your question, but let me just stop there.
Regulating cellular processes.
Of motion regulating cellular processes?
Utilizing the motion to regulate.
I don't know. You know, there's a lot about stress or stretch, but about motion per se. Certainly wound healing is motion, and that's what people in the wound healing literature measure. And even in development, there's a lot of measurement of motion. But I don't know, the answer to your question, I don't know. It might be known, but it's not known by me. It's a good question. Yeah.
Was there any cell proliferation during the...
Oh, good. Yeah. It turns out there's a lot, and about roughly half of the area expansion is due to proliferation in this case, about half. So there is proliferation, but even though there is, the stresses are mostly tensile, or they're almost all tensile. So you can't ignore the proliferation. It's there.
Before you remove the barrier, the cells are probably contact inhibited.
Yeah, that's right. Actually, we have data about that, and so the proliferation has gone down, and as you release it, then the proliferation, the cell numbers start to go up. Yes?
So the last few slides are on a relatively new study, but I was wondering if potentially the method of how the membrane was removed could affect how the cells begin to move.
Yeah, that's good. That's a good question. Actually, I don't know the answer to that. There are some other papers in the literature on wound healing who have asked just that question, because they say, is it the injury when if you actually, like scratch wound healing assays, you scratch the monolayer and you actually wound the cells, and then the question is, did the cells start to migrate because they're wounded, or did the cells start to migrate because there's free space into which they can crawl? Now, we don't really know if we're wounding the cells at the boundary. There's others who've shown that just creation of free space is sufficient to start crawling. Whether there's wounding here, in this one, it's a little hard for us to say. I guess you could figure that out if you knew more about injury than I do. But that's a really good question. But the way I think about this is that actually the cells are, if you will, they're jammed. They're so packed, cheek by jowl, that they can barely move up here. So they're just, the cell density is high, and when you create free space, there's a wave of unjamming, just like in a traffic jam. The light, if the light turns green, now there's free space, and there's actually a legitimate wave. I mean, a wave with a defined speed that starts here and moves back. If you look at the movies are online, you can actually see the wave propagate backwards, and as one cell moves forward, it makes space for the next one, and that propagates all the way across the monolayer. So is it a wave of unjamming? I don't know for sure, but that's what it looks like to me. Yes?
So there are many forces involved in all this. If we balance these forces and stop the cell from movement, then and when we just remove that force, will they start again to move with the same force or not?
If you stop them?
Yeah, stop them and then remove that extra.
Haven't done that experiment, although J. Kim is doing an experiment close to that where he's put a barrier in the way. So he's put a micropatent region where the cells can't crawl on, so they collide with the barrier, and we're looking at what happens there, and he's got all the data.
And after how much time they just lost this one? Like,
how much time?
If we put the barrier for a long time, that maybe they...
Oh, if you put the barrier, probably they're going to stop, and maybe you'll go back to a state kind of like this. But we haven't done that experiment yet, so that would actually be fun to do.
Do you get another wave when you put a barrier?
That's really interesting, because what this is, I don't want to get too techie on you, but if there's waves, it says there's something that's hyperbolic, it's a hyperbolic system. So if you put a barrier here, you might see shock waves or something going on, and we haven't actually looked for that yet. But I've been thinking about that maybe, I mean, from mechanically, this is a really rich system, and we're just scratching the surface, I think. So there's lots of interesting things to do. And actually, I've got a new fellow who just joined my lab, someone from Iran, actually, who had studied colliding plasma streams. And so she's setting up an experiment to have actually two monolayers actually run into each other. So it'd be similar to what you're talking about. Yeah?
Can you extend this approach to a three-dimensional?
I wish. It's just, that's a big and important question. This measurement technique only works in 2D. And so the question is, how much can we learn and how much will carry over to 3D? And the answer might be nothing or a lot. And that I wish I knew. There are lots of people studying how cells invade in 3D, and a lot of things are different, for sure. So don't know. On the other hand, two-dimensional systems, epithelium and endothelium are innately kind of two-dimensional. So that's kind of begs the question. So they're not irrelevant biologically, but again, this is a very reduced system. This is cells on collagen-coated gels, and just it is what it is. And if we can learn, but the notion of Plutotaxes, cells, wanting to minimize intercellular shear stress, that may carry over into 3D, or it may not, and that's probably testable somehow.
What about abnormal systems like, maybe setting up a monolayer of cancer cells that may have different cell-to-cell interactions?
We've done that. Yeah, that appeared last year. So we looked at, I can't remember, some breast cancer cell lines that have disrupted junctions, and it turns out Plutotaxis goes away. The cells lose their steerage. So we've done a little bit of that. Also, if you chelate the calcium, the cadhedrans go away, and again, this Plutotaxis gets ablated. So there is some data on that. Yeah.
So you said that the polyacrylamide gel floor that you were using is about 1 kilopascal. Have you tried looking at lower or higher stiffnesses and seeing how that? That's interesting. Yeah, we did. In the paper that appeared last year, actually a reviewer called for that. So we did experiments on widely different substrate stiffnesses, and we found no difference. Now, others looking at single cell systems and some have found big differences. And when we look at single cells, we find big differences. We published actually a theoretical and experimental paper, I think a couple of years ago, that if the gel is thin enough, and the monolayer is big enough, the cell looks virtually, the substrate looks virtually rigid. It depends on the width to depth ratio. And it could be these are wide enough that basically if you pull on the gel, you're feeling the rigid substrate, right? Whereas if you're one cell on a thick gel, you're feeling the gel, not the substrate. So anyway, we've got a paper on that we make the argument that it's the width to depth ratio, and these are effectively rigid, but those were not. It was not extensive studies. They were we did just enough to satisfy the reviewers. So I would call that an open question.
There's a paper by Mike Morrell with Paul Matsudaira on looking at epithelial cell collective migration on substrates of varying viscoelasticity. So it's
right.
That has a different texture.
Yep.
And there, one of the interesting things that he finds is that he sees these sort of swirling patterns and distances over which the motions correlate, and that changes abruptly as a function of stiffness of the substrate. So there's some interesting behavior.
Interesting behavior there. And then there's a recent paper that just appeared saying that PDMS and polyacrylamide are different. I don't know if you saw that. It's in Nature Materials. I can't remember who the first author was, but saying that if you match the material properties, same coating, but polyacrylamide versus PDMS, you get totally different behavior.
And maybe viscoelastic as opposed to elastic.
It could be that. I think they argued something about molecules being actually being mobile in polyacrylamide and not in PDMS. But anyway, so there's a lot of, the substrate is hugely important. I think everyone agrees on that. Yeah.
So have you tried doing similar experiments, but perhaps using a chemotaxis and viewing, see if
No.
there's any kind of wave?
We haven't. But that, it's going to be important to do, but that's not at the top of our list. But I mean, for people interested in those sorts of things, it'll be really important and easy to do.
Yeah, it'd be interesting seeing what the mass behavior would be. Chemotaxis, perhaps if the leading cells kind of guide that direction.
Well, they will. I mean, but then what are the force implications, the stress implications of that? Yeah, that's a really good question.
So is this collective behavior between the cell-cell adherent junctions and the cytoskeletal components, or mainly dominated by cell-cell adherent junctions?
Oh, which kinds of junctions?
No, the collective behavior.
Yeah.
Is dominated by the cell-cell adherent junctions, or is the combination of cell-cell adherent junctions and the cytoskeletal?
Oh, yeah, it's got to be both. We know if you take away the adherence junctions, the behavior totally changes. But those are connected to the cytoskeleton, so it's got to be both.
What if you do cytoclasm D treatment?
We didn't do that. We did do blebistatin to turn off the myosin motors. And this was an interesting thing in terms of those X waves I talked about. When you take away, this is actually, we don't understand this. So we thought that myosin has to be playing a big role in how the cells are spreading. So you use blebistatin, and when you do, the traction forces get knocked way down by more than an order of magnitude. The spreading velocity of the monolayer doesn't change, believe it or not. It doesn't change at all. So the myosin motor activity is reduced, but maybe the load against which it's pulling is also reduced. So the velocity of spreading is unchanged. That actually people had found that. So at first we thought it was crazy, but we found that others had found that. Interestingly, that wave that I talked about persists. You can still find it, but it doesn't move. It's stationary. You can find it just sits there. It's like a waterfall that doesn't, it just stands in front of you. So it's in the paper. You can read about it. We don't really understand that, and also the other thing is, if you take away calcium, it stops crawling. If you put the calcium back, it starts crawling again, and it launches another wave, which actually gets to someone's question over here about, is it an injury? So the answer is probably it isn't, because when we just put calcium back, you generate another one of these rearward propagating waves. So that wasn't injury, just bringing the calcium back. So it's, I don't know what's going on, but anyway, the measurements are now straightforward. So all these are answerable questions. Anybody else?
I have a question on the single cell mechanics.
Yeah.
The polarization happens in the tension only or in compression as well?
Good. Yeah. I was quite convinced that it would have to be both, because I was thinking about this in terms of molecules trapped in energy wells, and if you perturb them, you would then break the bond. We did the experiment. It turns out it happens in stretch. So stretch, return, you fluidize. If you compress and return, nothing. Compression does nothing, at least in the cells we look at.
So that cell rheology is mostly compression, and that's why you see this self-stiffening.
I'm sorry, I didn't.
Cell rheology, G prime, G double.
Yeah.
That's why it's consistent. You see stiffening there.
Yeah.
But
yeah. It must be that when you stretch, you have bonds that are under tension, and you can just basically break them. Or there could be severing proteins that are force dependent, and maybe you can expose regions that are more susceptible. With compression, we don't find any. Zero.
So with compression this way, you'd get extension this way.
You would.
But I guess if you've got all the stress fibers in this orientation.
They're not totally in the plane, but they tend to be close. So yeah, anyway, that's what we find. And actually, I think we published that somewhere.
Is there any threshold force observed?
No, it doesn't seem to be. It seems to be like the more you stretch, the more fluidization you get. And in the cells we study, you can knock with a 10% stretch, you basically can knock the tension down to about 0. And with smaller stretches, we haven't really looked hard for thresholds. So, but at least in the range we've looked, it just seems to be the more stretch, the more fluidization. But I don't really know. Yeah.
Just one question. The experiments you have performed is more or less like expansion of the cell sheet into empty spaces.
Yeah.
But for example, during the development of the embryo, you don't really get a free space.
Right.
So,
right.
How do you think, for example, Plutotaxes will change in this case? Because from videos from Ray, I saw some like shearing of the cells,
yeah.
movement, like lateral movement of the cells.
Yeah.
So in that case, is like what you're saying, is it violated or is totally
Yeah.
different?
Well, I wish I knew. I mean, every time I think about development or hear talks like that, this one was actually superb. I get a headache because I can't. It's so much more complicated than these systems. I wish we knew the answer. So this is a very reduced system as compared to like a fly gastrulation. So some of these things probably come into play, but it's a different system, and I just wish I knew, but we're not there yet. On the other hand, there's a guy Schwartz, Martin Schwartz, who's now at Yale. He now has probes that can actually measure forces at cell-cell junctions, and maybe doing that in the fly, one could take these, get maps like these, but in a more biological system. Maybe that's coming.
Jeff, maybe that's a good place to stop.
Oh, okay.
You're welcome to join us for lunch for further questions.
Okay, yeah, I can hang around for a little while.
And just over here. But thanks again.
Great.
It's fantastic.
Oh, thanks.
And that the jump, we're